academician Dr. Thanos to welcome for his welcome remarks and his opening remarks for this event. Thank you.
Good afternoon. Distinguished guests, the former General Secretary of Higher Education and former President of the Hellenic Open University, Professor Odysseas Zoras, is not here to actually greet him. Dear faculty members, wait, she just came? Oh my God, this is amazing. So, well, I'm particularly happy since he made it right at the right moment, at the right time. Dear faculty members, colleagues, and most importantly, our prospective students, it is with great pleasure to welcome you to this milestone moment, presenting our new graduate course, Personalized Medicine and New Therapies, here at the Biomedical Research Foundation of the Academy of Athens, in collaboration with the Hellenic Open University. Today marks the beginning of an exciting journey, one that I'm confident will shape not only the students' academic pursuits, but also their professional and personal growth in profound ways. This graduate program represents a bold step into the future of healthcare and biomedical research, a future where medicine is no longer one size fits all, but instead today's medicine is tailored to the unique biological, genetic, and lifestyle profiles of the patients. We are living in an era where groundbreaking advancements in genomics, proteomics, biotechnology, epidemiology, and data science are converging to redefine how we approach treatment, prevention, and care. This graduate program has been carefully designed to prepare the students to be at the forefront of biotech in genomics, proteomics, biotechnology, epidemiology, and data science. And the students to actually participate in this ongoing transformation to meet the evolving needs of a dynamically changed society. Precision medicine is not just an emerging discipline, it is a paradigm shift. Its promise lies in its ability to optimize therapeutic outcomes, minimize adverse effects, and offer hope where conventional treatments fall short. From cancer immunotherapy to gene editing, and from pharmacogenomics to advanced diagnostics, the field is reshaping what we thought was possible in medicine. We are particularly proud of how this course integrates knowledge and tools to navigate and contribute to this exciting landscape. The key highlights of the course of the program include an interdisciplinary approach where we provide blending insights from genomics, genetics, pharmacology, proteomics, data analytics, and clinical practice. Hands-on learning opportunities through case studies, lab research, and collaborations with leading experts in the field. Cutting-edge technologies, covering advancements like CRISPR technologies, mRNA therapeutics, artificial intelligence in drug discovery, and more. Ethics and policy discussion, this is a very important issue, addressing the societal, legal, and ethical implications of personalized medicine and its accessibility. But let me remind you that graduate education is not just about gaining knowledge. It's importantly to understand that it is also about creating this knowledge. It's about questioning the norms, seeking solutions to real-world problems, and contributing to the larger body of understanding that will benefit the society as a whole. I would like to take the opportunity to extend my heartfelt thanks to all who have worked tirelessly to make this program a reality. Special thanks go to Professor Odysseas Zoras, the former president of the Hellenic Open University, who actually conceived this program, and he proposed this collaboration with the BRFAA. The Hellenic Open University professors, Maria Hadjinikolaou, Argyros Guru, and Achilleas Kameas, and my colleague at BRFA, Professor Dimitra Mangoura, whose vision and hard work have made this program possible, and of course, our dedicated faculty and administrative team. Their hard work and dedication have made this day possible, and their efforts will undoubtedly leave a lasting legacy. Once again, I welcome all of you to the Personalized Medicine and New Therapies graduate program. Together, let's make this an enriching and impactful experience. Thank you.
Thank you very much, Dr. Thanos, for your warm and meaningful words. And now I would like to invite the rector of the Hellenic Open University, Professor Achilleas Kameas, to extend his remark to all of us. Dr. Kameas, please.
Good afternoon, everyone. Thanks for arriving at this moment. First of all, a correction. I'm not the rector, I'm the Dean of the School of Science and Technology, but I think that our rector will agree with the importance of this new graduate course that we are launching in Hellenic Open University. Dear Professor Zoras, our very dear ex-president of Hellenic Open University, dear Professor Thanos, dear Professor Mangoura, dear Professor Hadjinikolaou, dear Professor Zgourou, let me thank you for all your efforts for making this program a reality. It is with great pleasure, and I would say with also great joy, big joy, that I am participating in this event. Unfortunately, I cannot be there with you in person, as I am representing Hellenic Open University in a very important meeting in Barcelona, the kickoff meeting of the European Universities Open EU Alliance, the alliance of all European Open Universities towards the establishment of a European Open University. It's a very important event, and again, thanks to Professor Zoras, we are in this group of universities. The graduate course in precision medicine and novel therapies really is going to change the map of competencies and occupations in Greece. It's a very innovative graduate course that Hellenic Open University, and especially the School of Science and Technology, is proud to launch together with the Biomedical Research Foundation of the Academy of Athens. The scientific coordinator the director, I would say, of the graduate course, Professor Thanos, already spoke about the contents of the course and the aims. Being a top expert on the field, I think he conveyed in an excellent way where we started from and where we want to arrive, what we want to achieve with this really important program. The School of Science and Technology, as always, is at the forefront of science and of technology. This program is an excellent demonstrator, a proof that we are always looking to move to push education in Greece beyond the current limits towards the international state of the art And this we do very well in this Faculty of Science and Technology of Hellenic Open University. At the same time, this program marks a new orientation in our school, in our faculty, opening towards medicine and the relationship of this very important discipline with technology that we serve in our faculty. So, having arrived at this point, all I can do is wish a big success, of which I'm really certain, to this new program. Congratulate and thank you again for laboring so hard to bring this program into fruition, to give birth to this program. And we will be here as a faculty and also as a university to support it, to promote it, to put in practice all the new methods and tools that we are using in our university, which has in the past 20 plus years proved that they are really accepted by our society and have, thus Hellenic Open University has brought a big societal change. So as HOU was a paradigm shift in the education in Greece, this new program is going to be to bring into our society a paradigm shift in modern medicine. Looking forward to giving the first certificates, the first degrees to the graduates of this program. Congratulations again and thank you very much.
I cannot hear anything. I heard Achilles, but I cannot hear anything from the room. Yes.
Okay.
Now I can hear you. Fine. Please repeat.
Okay. I just want to thank Dr. Kameas for his nice remarks. And now I would like to ask Professor Panagiotis Kagialis, the Vice Dean of the Hellenic Open University, to give us his remarks on this special event. Dr. Kagialis, please.
Yes. Thank you very much. I will be very brief as unfortunately I cannot be with you today as I am participating in doctoral dissertations defense right now. So I came out just to be with you briefly. Dear colleagues, dear prospective students, ladies and gentlemen, I have the great pleasure to share with you the warm welcome of our rector, Professor Manolis Koutouzis, who unfortunately cannot attend this meeting as he's on a flight to Barcelona to attend a major event for European distance learning universities, the one Achilles Kameas just referred to also. So in my capacity as Vice Rector for Academic Affairs and Outreach of the Hellenic Open University, I must assure you that we are very proud and indeed honored to participate in the new master's program in precision medicine and novel therapies, which, as I'm sure you're all aware, is jointly offered by the School of Science and Technology of the Hellenic Open University and the Biomedical Research Foundation of the Academy of Athens. We are indeed eager to see this cutting-edge program flourish and are confident that it will set off successfully in the following weeks. To this goal, our rector has actually named PREH as one of our 5 so-called hero programs in the context of our campaign for this semester's 40 program offerings, which means that this particular program is being currently heavily advertised in a variety of media. So please allow me to extend my own cordial thanks to the program's steering committee for organizing this important event, and wish you all a successful and enjoyable learning experience in the following hours. Thank you very much.
And thank you, Dr. Kayelis. I would like now to invite Dr. Zoras, the mastermind of this program and the previous president of the Hellenic Open University.
Yes, so thank you very much for your kind words. Ευχαριστώ πάρα πολύ όλους που σκεφτήκατε να με καλέσετε. Διότι συνήθως γίνεται η εναρκτήρια τελετή, γίνονται όλες οι φιέστες κλπ. Και έχουν ξεχαστεί αυτοί οι παλιοί, έχουν φύγει, έχουν ξεχαστεί. Και πραγματικά έτσι πρέπει να γίνεται, γιατί το μέλλον ανήκει σε αυτούς που αναλαμβάνουν να τρέξουν τη δουλειά. Χαίρομαι που βλέπω τον Αχιλλέα, τον Καμέα, καλό φίλο από το παρελθόν. Χαίρομαι που βλέπω τον κύριο Καγιαλή, την κυρία Ζουγρού, την κυρία Χατζητικολάου, τους συναδέλφους από το ΙΒΑ, τον καλό φίλο τον Δημήτρη, τον Θάνο. Και είναι το αποτέλεσμα, είναι προδεδικασμένο με την έννοια ότι 4 ισχυροί χώροι θεσμοθετημένοι, 2 μεγάλα ιδρύματα όπως είναι της Ακαδημίας Αθηνών και το Ανοικτό Πανεπιστήμιο συνεργάζονται. Συνεργάζεται πολύ γρήγορα, θα έχουμε και το κτίριο που αφιερώνει η Ακαδημία στην εξατομικευμένη ιατρική, ένα ολόκληρο κτίριο, το οποίο θα είναι στην υπηρεσία προφανώς και αυτό του Μεταπτυχιακού και φυσικά και το εργαστήριο βιοασφάλειας στον ανώτατο βαθμό που έχουμε στην Ελλάδα, του κυρίου Θάνου. Όλο αυτό συνθέτει ένα σκηνικό, το οποίο είναι σκηνικό επιτυχίας. Οι άνθρωποι, τα ιδρύματα, τα institutions τα οποία θα εμπλακούν είναι συνταγή επιτυχίας. Συνεπώς, αποτυχία δεν υπάρχει, η επιτυχία είναι προδεδικασμένη. Και επίσης θέλω να πω ότι και στην εποχή της τεχνητής νοημοσύνης θα μπει και η τεχνητή νοημοσύνη στην εξατομικευμένη ιατρική. Προχθές συζητάγαμε με συναδέλφους γιατί πολλές κλινικές μελέτες δεν επιτυχάνουν. Πολλές κλινικές μελέτες δεν επιτυχάνουν γιατί είναι τόσες οι μεταβλητές παράμετροι των ασθενών οι οποίες μπαίνουν μέσα που είναι αδύνατον να γίνει μια ταξινόμηση ορθολογική. Και μόνο με την τεχνητή νοημοσύνη αυτή η ταξινόμηση θα μπορέσει να λάβει υπόψη όλες αυτές τις παραμέτρους, ώστε οι κλινικές μελέτες να αποδώσουν το μέγιστο που είναι δυνατόν. Συνεπώς, μια συνταγή επιτυχίας. Ευχαριστώ όλους τους φίλους, κυρίως ευχαριστώ τους ακαδημαϊκούς, το περιβάλλον όλων των ακαδημαϊκών, που και τον πρόεδρο του ΙΒΑ, τον κύριο Λουκά Παπαδήμο, ο οποίος σε ανύποπτο χρόνο είχε ευήκονους και συνέλαβε την ιδέα και συνέλαβε ότι αυτή η συνεργασία θα είναι καλή και αποδοτική. Συνεπώς, όλοι μαζί θα κάνετε ένα εξαιρετικά καλό έργο και κάποια στιγμή φαντάζομαι ότι θα με θυμηθείτε πάλι και θα με καλέσετε όταν δώσουμε τα πρώτα πτυχία. Ευχαριστώ πάρα πολύ. Βλέπω και την κυρία Γετράκο, η οποία είχε συμβάλει πάρα πολύ οργανωτικά στην αρχή της όλης διαδικασίας, διότι και το οργανωτικό κομμάτι δεν πρέπει να το αγνοούμε. Είναι σημαντικό. Συνεπώς, ένα μεγάλο ευχαριστώ. Η συνταγή επιτυχίας είναι δεδομένη και σας εύχομαι καλή επιτυχία σε όλους. Ευχαριστώ πολύ.
We would like to thank Dr. Zoras for his encouraging words. We need them. And now we will go on with the scheduled program with the presentations of the program and the modules. And I would like to start with a PowerPoint presentation. Okay, and our first speaker will tell us all about the innovative research that we're doing in Brafa. Dr. Thanos, please.
Good afternoon again. So I'm going to give you a brief overview of what is going on here at BRFAA, and you can actually formulate in your mind how exactly our activities can help in the implementation of this graduate course. So, so BRFA is the youngest, is the most recent, let me use this wording, research center, biomedical research center in Greece. Is only 23 years and has a variety of goals, but all of them converge in one thing, is to actually achieve excellence in research, in biological, in biomedical research, in education, and in providing high quality health services. So the idea of creating BRFAA, this was Professor Skakea's idea, was a remarkable and highly innovative idea nearly 40 years ago, when he actually conceptualized that if we can manage to actually implement and integrate basic research, basic biological research and clinical research under the same roof, this will provide a unique opportunity to actually provide a high quality and have ready high quality biomedical applications. And don't forget that basic research is actually the fuel to actually have a really very rich economy of knowledge. And on the other hand, there is no applied science if there is no science to apply. So although Skakeas was a surgeon, he had this broad view of science in a worldwide manner. So the BRFA's overall scientific mission is shown here, and which actually shows all these different approaches which are being followed in our institute, which actually aims at determining the molecular basis of processes of health and disease, and turning this knowledge to actually improve the quality of the health services provided to the patients. So the missions are shown here. So it's to perform excellent basic research in biomedicine, to implement translational research and precision medicine in Greece, to offer vital services to scientists in Greece and abroad, and to the public. BRFA has unique infrastructure which actually support many research projects, not only in this institute, but also in the entire country. Trains students and scientists and visitors at all levels to actively engage in technology transfer and industry relations, and of course to coordinate and integrate life science research through the implementation of emblematic projects. Now, there are 6 thematic areas in the Institute now. There are 6 main topics which are being investigated. And you see those here. We have a cardiovascular research, cancer, immunology and inflammation, neurosciences, artificial intelligence and molecular epidemiology, and of course, stem cells and regenerative medicine. All of our investigators are actually working in at least in one of these horizontal thematic areas. On the other hand, the Institute has an internal vertical distribution of activities. You see those here from for the Center of Basic Research, where we actually approach all these topics by doing purely basic research with some applications, and of course the Center of Clinical Experimental Surgery and Translational Research, which actually uses all these technologies and all this new data generated from this direction to this direction. And of course, the Center of Systems Biology, which actually focuses in trying to formulate by generating algorithms and also the artificial intelligence tools to trying to understand some of these basic biological concepts. And the Center of Environmental Effect on Health, which is actually underdeveloped at this time, but we look forward to further developing this, taking into account the ongoing climate crisis. So one of the very important aspects and one of the very important things which actually separates and distinguishes our institute from nearly all others is this triangle here, which is a dynamic flow of information either direction and to any of the two partners. So it's for research where we actually ask important biological questions which to be answered require the development of new technologies which actually can give rise to the provision of nearly novel services which produce economic wealth, which actually is being reinvested into this institute to further advance the infrastructure. And this infrastructure is shown here. This is the Academy of Athens building with the 6 pillars. And we have developed and actually continuously renovate all of our technologies, further advancing these technologies from genomics to biobanking, bioimaging, a state-of-the-art animal house, proteomics, and clinical unit. There is a clinical unit in this institute where we can actually carry out clinical trials, and this is and we have renovated an entire wing at the Sotiria Hospital with 32, I think, beds, a completely new wing where we can actually carry out clinical trials in collaboration, of course, with the hospital, with the Sotiria Hospital. And here you see some of these unique infrastructures. This is a semi-intensive care unit which exists here in this building, which is used for cardiological measures. The experimental surgery unit, the PET-CT. This is the only institute with a clinical PET-CT. Of course, this is the hospital wing at the Sotiria Hospital, which we have renovated and which is ready to be used for clinical trials. And of course, the state-of-the-art animal house, a multiphoton microscope, which was installed a year ago, and of course, the Hellenic Blood Bank, the only one in Greece in a public research organization. And at the center of all of this is the Greek Genome Center, the first and unique in Greece genome center, which actually can carry out high throughput genomics analysis and has played the most important role during the COVID-19 pandemics. And because of its central position and state, the Greek Genome Center has collaborated with all research institutes in Greece, nearly all hospitals in Greece and abroad, the research centers abroad, and of course, with private companies where it provides services and brings some economic wealth to BRFAA. Now, BRFA has established a very strong and very dense collaboration networks. You see those here, either regional with nearly all Athens research and health providers, a national network with, again, all Greek universities and research centers, and with institutes abroad, where some of those, you see some of those here, but it's much more optically amazing. You see the interactions between our investigators with all other institutes in Greece. You see a very dense network of interactions. Now, scientifically speaking, and using this, using metrics, from 2018 to 2024, there are about nearly 2,000 publications in international journals, of course, where you see that BRFA has a very, this very dense network of interactions is translated to really highly collaborative interactions and publications, which actually are depending on grants, which the investigators have actually succeeded to recruit to BRFAA. And you see, for example, this for 2023, I don't have yet the cumulative numbers for 2024. For example, 111 grants were active in 2023, which from the European Union, from GSRT, the executive, private, and one from the Ministry of Interior. You see all these grants which actually provide the fuel for research, technology, innovation, and of course all of those are translated to biomedical applications and services. And the personnel for 2024, 407 people of all different levels, from the directors to a graduate student and a master's student were actually working at the institute, and as you see, women are actually the majority. In fact, I should use men are the minority. This sounds better. So we consider ourselves the largest biomedical research training school in Greece, research center center where nearly 200 PhD theses have been completed up to 23, ongoing PhD theses and completed master theses, and ongoing master theses have been actually ongoing because a lot of students are coming here because of the infrastructure and the willingness of the personnel of the research personnel of our faculty to actually provide all this guidance which is required to either complete to carry out a PhD thesis or a master project. And of course, we participate in 51 graduate programs in collaboration with academic organizations in Greece. And you see here, you have a view of with nearly all medical schools and nearly all universities in Greece. We collaborate with those, and we actually have succeeded to build this very dense network, as I said, which we think is very important. However, our collaboration with the Hellenic Open University is unique because it's the only one. It's the only one of these collaborations where BRFAA plays a very crucial role in actually design the graduate program in collaboration, of course, with the faculty at the Hellenic Open University, and this is what we have done. You see that again because shows all these interactions which are being formed within BRFAA between the different programs, as I said, which all of them, the heart of this program at BRFA is to achieve excellence in in science, in training, innovation, and in providing services. Those are very important issues because we live in a world where the only standard thing is change. If you don't change, if you don't develop further, if you don't evolve, you die. So as I said, we provide biomedical services, which actually provide a very nice field, a very nice landscape where young students, graduate students, master's students can be trained from the unit of the clinical trials to the diagnosis and prognosis and to the development of novel therapies in collaboration, of course, with the clinical units. And of course, as I said, all of those are really very, very well synchronized in a manner where we can actually achieve measurable results. So there are new investments because, as I said, change is the only constant thing in life. So, as Professor Zoras mentioned, we actually in the process of constructing the new BRFA Precision Medicine building. There is under construction the Biosafety Laboratory 3+, a comprehensive imaging and radiogenomics laboratory. Those two are being funded, the last two are being funded by the RRF, the Recovery and Resilience Fund from European Union. We established collaborations with research and academic organizations on data science, artificial intelligence, and machine learning, because we think that, as Odysseas Zora said, artificial intelligence will be one of the main tools for the future. We implement the Genome of Greece project. I don't have much time to talk about this. This is an emblematic thing in the context of the Genome of Europe, which is the emblematic European project. And of course, we upgrade and expand the BRFA biobanks. So we will focus on precision medicine in the cardiovascular diseases, in cancer, neurodegenerative diseases, immune and infectious diseases, and actually develop technologies, new technologies for stem cells and regenerative medicine. Now, one of the very important things is, as I said, the BSL-3 laboratory. So this is under construction. The laboratory, which will be collaborating close in a dynamic equilibrium with the pharmaceutical industry, with the National Public Health Organization, with university hospitals, and with hospitals, because we think that public health is a very important component, which BRFA has a very strong very strong immunology group for basic and applied research. So we will try, and you will hear about that immediately after, and we will try actually to exploit all this new knowledge, which is produced nearly every day in here. And of course, you see the BSL-3 Plus lab. It's a unique infrastructure, which we think that's going to make a difference in Greece and will significantly advance our concept of public health, and they will put Greece at the forefront of research in Europe, because there are very few such laboratories in Greece, and we are happy that the government agreed actually to fund this in the context, as I said, to the RRF, to the Resilience and Recovery Fund, and of course is being co-funded by the European Investment Bank. So this is my last slide. So as I said, our vision is to be the top research centers in Europe, in research, in education, in training, in precision medicine, and in health services, and in providing health services. So with this, I would like to thank you, and I'm sure that the talks, the presentations which will follow will further enlighten our understanding in precision medicine. Thank you.
Thank you very much, Dr. Thanos, for this very informative presentation of our activities. And I would like to ask Professor Maria Hadjinicolaou from the Hellenic Open University, a member of the steering committee of this program, to give us the essence of a student-centered education.
Thank you very much. I also welcome you. Thank you for being here. Following the introduction made by the Vice Rector and the Dean of the school, I will skip the presentations regarding the Hellenic Open University and the school facilities infrastructure. And I will start, I would like to, in my talk, to present the mission of the Hellenic Open University, explain what is the student-centered education we offer, and show how this is in line with the design of the new master's program regarding the precision medicine and novel therapies. So, the mission of the Hellenic Open University is the provision of undergraduate and postgraduate education and training, and the advancement of scientific research and technology through distance learning methodologies. Hellenic Open University aims to attract wide audiences of different ages, professions, environments, countries, and committed to lifelong learning values. Furthermore, Hellenic Open University, at this new era of university, starts to explore and strengthening collaborations with universities and research institutions such as the BRFA. What do we mean when we say that it offers student-centered education? We mean that there's no entry exams. Each student can keep his her own self-space and study, and following the different learning style that can have. They can study, students can study from home or from any place, so there are no classrooms that have to be there in some particular place, and can study anytime through the asynchronous mode of education. Students are divided in small groups and are guided by tutors that they give them support through the whole journey of their studying. The program, which is something new and make difference from the other standard universities, that we consider the program as a set of modules, which means that one can enroll only to one module each time. The students have to follow a weekly calendar where education material, learning activities, and projects are allocated for each week. But now I would like to give some reasons and to describe why this program is unique and what are the strengths of this program, and what is the design, the philosophy behind the design of this program. This program this uniqueness and strengths of this master program lie in the synergetic cooperation of Hellenic Open University and the Biomedical Research Foundation of the Academy of Athens. The Hellenic Open University brings expertise in distance learning, flexibility, and a robust administrative mechanism, educational infrastructure, and worldwide academic and social reach. The BRFA is leading research institution in Greece, specializing in precision medical research. This partnership guarantees a more successful operation of this master's program within the distance education framework. It requires and also provides interdisciplinary knowledge. It is organized as a set of modules, as I mentioned, which are focused on the most important aspects of precision medicine you heard before. It addresses the new challenges in the fields of molecular profiling, diagnosis, clinical trials, and advanced therapeutics, as Professor Dr. Thanos mentioned before. Combines with something new in distance learning programs, theoretical knowledge and hands-on laboratory experience, allowing also to perform a master thesis by research in the state-of-the-art facilities and laboratories of the BRFFA. This opportunity enriches the distance and lifelong educational experience, while providing a pathway for those who seek to pursue an academic career. Moreover, the tutors are all scholars with outstanding research backgrounds, and this is motivating the students that are connected with them and interact. So, the question of people who are already communicated with us and ask what are the qualifications that someone who graduate will acquire. So we mentioned some here. Students that graduates will be able to promote health and manage patient care, evaluate laboratory data and biomarker assessments, participate in clinical trials and develop new therapies, utilize AI, machine learning, and computational methods. Moreover, they will be able to write and engage in scientific communication. This is the slide you can follow and have more information about the program and all I mentioned. Hellenic Open University, the programs have already certified by the Hellenic Authority of Higher Education. Here are some pictures of the certificates. Hellenic Open University, as a member of international organizations regarding distance teaching and distance learning, like the European Association of Distance Teaching Universities and the International Council for Distance Education, knows and updates the know-how on all aspects regarding and concepts regarding distance education. Here's some useful information regarding the enrollment requirements. For graduates with those are accepted from schools of medicine, pharmacy, and biology, from the part of engineering, what kind of engineering, information technology, computer science, and computer science with biomedical applications. So it's kind of saying that why you are trying to expand yourself to medical aspects and medical concepts. No, medicine is coming to science and technology, so that's why we are here to collaborate and improve the knowledge and be useful to society. So English also are very is demanding, it's high requirement because all teaching material and communications are in English. The tuition fees, so you have the program is structured in 2 academic years and follows 2 distinct directions. The European credit transfer units are 122. The program consists of 5 modules that you see the cost per module, and they have some different prices for different kind of laboratory modules. But the 2 structures are here. So this is the first structure, the cost for one from the structure A and the structure B. You will be here and from following presenters and the exact structure of these 2 directions of study. And here is the site for people who are attending and want to enroll. Admissions are open. Here is the website. You will see in front. You will see this is what this picture, and then you have to just to apply for the program after having here all this today. And also, if you would like to go through the website of the university of the program and have some info. Everyone is here to help you and either with mail and telephone and help you to apply for the program. Thank you very much.
Thank you, Dr. Hadjinicolaou, for giving us the core principles of this whole endeavor and some really good practical information. And I would like to now to ask Professor Argyros Gourougou, also a member of the committee, also a member of the steering committee, to give us some more information about the distance learning environment and the Hellenic Open University platforms. Dr. Gourougou, please.
Thank you very much for welcoming me in this meeting, on this open day today. And since we are speaking about distance learning, this is an attempt to navigate through the educational platforms that are supported from the Hellenic Open University and see some very basic features and familiarize with them. As soon as someone types courses.app.gr, you may enter to this environment. This is a Moodle platform. I hope you will find it very friendly as we do. Every of these modules of the program have its own space in the platform. So in order to enter one of the modules, you have to click on a blue button. Here, I need to say that I'm sorry for the Greeks on my slides. The platforms are totally translated in English and will be ready before the program starts. As we enter within a module, a specific module, what you can see, first thing we have to observe is the calendar. The calendar is monthly based, and there are highlighted dates on the calendar. The pink one, can you see them pink? The pink one are referring to some obligations you have on educational activities, and the yellow ones are referring to the group counseling meetings, which can take place only on Saturdays or Sundays, only weekends then, and have a duration of about 4 hours each. What else can we do? Can we see? We can see communication days and hours for the director of the program, the coordinator of the module, our tutor, so we can have the teaching staff. And someone can have some personal communication with them. Going further, what else we can see? How a student is evaluated through the semester. We have some written assessments, 2 written assessments, which contribute to 15% each of the final grade. How is an assessment like this? A written assessment. This is a paradigm, an example from another program. So someone have to answer all the questions on the written assessment, upload it on the platform so the tutor can find it, correct it, grade it, and return to the student with some comments, some personal comments. Another educational activity is for our self-assessment and contributes to 5% of the final grade. Often there are quizzes that the students have to answer and submit to the platform, which is automatically corrects them so one can see their grade. Going further down, we can see the study weeks. There are 14 study weeks in each semester. And let's see what we expect, what is included within one of these weeks. Let's say week 10. In here, we can see the proposed chapters from the educational material, proposed scientific literature, video lectures, and whatever our tutor believes it's important for the student to follow the education, their study. How we can access this educational material? There is an e-reader supported again by the Hellenic Open University, which opens this material in a new window where someone can access this material. Either it is connected, he is connected or not connected to the internet, but cannot print it, download it, or send it to anyone else because it is protected. Also, you may see the learning outcomes referring to this week's educational material. Last thing I will mention, there are discussion forums. Discussion forums are separated in a forum where our tutor or the director of the program or the coordinator may have some announcements or messages for the students, like this one, let's say. And there is another separate part of this forum where the students can talk or communicate each other. The group counseling meetings, which are held, as I said, on the weekends, are supported by the WebEx platform, where the student can have camera and microphone on and communicate with the tutor and the rest of the students. And now the exams. Exams, there is another platform, the exams platform, where someone can find guidance, how can navigate this platform, or there is a demo to familiarize our students to the exam method, and the whole platform is as friendly as the educational platform. The exams contribute to 60% of the final degree of each module. So usually there are multiple choices where the student has to complete in a specific duration of time, and followed by oral examination by their tutors and on the WebEx platform. So these are the basic things. Now I wish to thank you for your attention.
Thank you, Dr. Zgourou, for this very meaningful navigation through the system. And now I will continue with me. Let me introduce myself very briefly. Professor Dimitra Magoura, Mimika Magoura is better. I'm Vice Director of the Basic Research Center of Braham, and I would like to go a little bit, if I can, deep into what really precision medicine is all about and why this program is so important and so timely organized. So precision medicine is an approach, is the most novel current approach for disease treatment and prevention for both. And what it really does is that it takes into account individual variability as we know it from the analysis of genes, environmental impact, and lifestyle for each person. So this type of approach will allow doctors and, of course, researchers to predict more accurately which treatment and prevention strategy is best for an individual. It's actually in total contrast with what we practice till today, which is one-size-fits-all type of approach, and with not much if there are any differences, up to only the physician, the practicing physician judgment to call differences. I would also like to make a distinction between the term precision medicine and personalized medicine. As a matter of fact, the two can be used for the same person, although the International Initiative for Precision Medicine is a little bit worried that if we say personalized medicine, we mean that we will develop for each one individually a treatment. No, what we're doing now and what precision medicine is doing now is subgrouping diseases, so that when we eventually will see a person, we will know to what subgroup the person belongs. This is a nice schematic which shows the single treatment, one-fits-all type of thing. And if you give this one medicine that we have, you are going to take very different effects. Some will respond, some they won't, and some they will do fine. So what truly precision medicine wants to do is to come to the personalized medicine, whereby after genetic analysis, biomarker analysis, and all the data we can get for lifestyle and environmental impact, we're going to know this, we will have a good subgrouping of the disease, so we will know which drug to give to each subgroup. You can call this one hypertension, you can call it cardiac failure, you can call it whatever you want, but there are subgroups within each disease. So what really precision medicine does is that it describes the disease, the definition of a disease at a higher resolution level by using all these novel technologies, and it is trying to enable more precise targeting of subgroups of the disease. So I'll give you an example here of how just simple genetic analysis has changed the landscape of the diseases. So until 1993, we thought that neurofibromatosis or von Recklinghausen disease was only one. Yet when genomic studies show that we really have three different diseases. So it's neurofibromatosis type 1, the gene is in chromosome 17. We have type 2, the gene Merlin is on chromosome 22. And then Legius syndrome, where the spread gene is on chromosome 15. But as I told you, now we have advanced enough to take into account many more things, and not just genomics, because we know that the genome can be modified. So we have to know the epigenome and what this altogether express. That is, we have to know the protein, the total, we have to know what kind, the population of proteins a sample or a person has, the transcriptome and the metabolome, and you will hear more about that. So we need this omics, but we also need lots of social and behavioral data. Simplest example is smoking. We need to know, is the person smoking, is overeating, stuff like that. Or we need to have the knowledge and the data for environmental factors. What is the climate the person lives, and so forth. And once we have all these tons of data, we need to analyze them with big data analysis in order to come up with a strategy plan. So what really precision medicine is deep phenotyping of diseases. So we need to know, if I could find this, right? We need to know all these things about each person of the group and then analyze it so we can generate the subgroups I told you about with big data analysis tools and integration, come up with a strategy plan, and not only treat, but make sure that the person we can prevent disease. That's very important. So, of course, artificial intelligence for this part is going and it's playing more and more important role. So how do we come up, how we can get to this magic bullet here, the magic drug? So I'll give you a workflow example for precision medicine for cancer patients. So first of all, we need to collect samples of data from the patients. And then we have to do molecular profiling using omics techniques, collect all these lifestyle and environmental factors data. And this all together must undergo computation analysis, where target and biomarkers will be identified, and most importantly, drug candidates, including repurposing of drugs. So once this is done, we can generate a treatment plan, but then what? Then we have to do testing of this treatment, and we can do that in vivo, in vitro, in silico, and of course in clinical trials. So when we have all that, the doctors will have a wider ability to use patients' genetic and other molecular information as part of routine medical care. The key word here is routine, okay? So greatly improve the ability to predict which treatment is best for each person. We are also going to gain a lot of understanding of the underlying mechanisms by which diseases occur. And as I told you, we are going to improve prevention, diagnosis, and treating of a wide range of diseases. And this is also something we don't think about, but it is as important. We will have better integration of electronic health records in patient cares. And imagine that, especially in a country like Greece, with all these islands, how important it will be to have all that. So what are the immediate hits for advancing precision medicine? Of course, the research and the efforts we all do. But actually, the most important part is that we have to prepare the next generation of scientists, which are going to be responsible for creating. I'm not going to mention every step, but you understand that in the end point is to create new diagnostic tests and new tailored therapies to aid earlier diagnosis, optimal treatment, and prevention of diseases for people. So this master program, the Precision Medicine and Novel Therapies, has been designed to serve exactly this purpose, okay? So it covers all the major aspects. It's going to give the know-how for all major aspects of what is needed to serve precision medicine. And you are going now to hear the modules that were carefully designed and developed to serve exactly this purpose. So you will hear, this is in alphabetical order, but you are not going to hear it like that. Applied omics, bioethics in clinical trials, bioinformatics for precision medicine, biomarkers in precision medicine, biomedical, clinical, and translational research, computational biology, and applied statistics for precision medicine, and molecular and clinical aspects of cancers. Finally, this is kind of the novel therapies we already have, but also how these are going to be enriched towards clinical application. So also, all this, you will see that they are nicely connected, and they absolutely cover, as I told you, the most important aspects. So that's what I have to say. You can find us at the website if you want to see more specific details about the instructors of this program. All of them, or the vast majority of them, are investigators here from Programma. And with that, I will give the stage to my colleague, Dr. Kostas Vekrelis, who will present the first module.
Thank you, Mimika. Can you hear me? No? Yeah. Okay, thank you, Mimika, for this informative introduction of personalized medicine and its aspects. I'm Kostas Vekrelis, and together with Eleni Katsadoni and Konstantinos Stamakopoulos, we've built up a team to teach the module of biomarkers in precision medicine. So simply put, biomarkers are measurable substances in an organism whose presence suggests a normal or pathologic state, or a disease state, or a response to an intervention, usually a pharmaceutical intervention. Biomarkers can include a wide range of molecules and characteristics, and they can be proteins, genes, and imaging findings, amongst others. They are very important in tailoring treatments to individuals, considering they help us to improve how well we can predict a person's risk for a disease, how a disease might progress, the staging of the disease once it's diagnosed, and of course, how an individual will respond to a medicine and the side effects of mainly drug treatments that they're undergone. I thought I would take an example from a disease which is very close to the disciplines I study, and this is neurodegenerative diseases. So I thought I would just give you an example of biomarkers in Alzheimer's disease. And you probably know it's a progressive neurodegenerative disease, a dementia, in other words. We know it's characterized by depositions of two proteins, amyloid beta and tau. And these depositions, these pathologies in the brain, start in different areas, but most importantly, they can precede any symptoms by 30 or so years before diagnosis is given. So we now are in a position, because of the development of biomarkers in neurobiology field, to overcome this misdiagnosis of patients with Alzheimer's disease. We know there's about 20 to 30% of patients that are misdiagnosed. They have another disease and they're misdiagnosed for this. And why do we do that? We have biomarkers, which are amyloid beta and tau protein levels in the brain, and we can detect them not only in the cerebrospinal fluid of these patients, but also in their blood. We know that this ratio will give us the staging of the neurodegeneration, the progress that I said before of the disease. But further, we know that with the help of omics in biomarker development, we know that genotyping for ApoE will also tell us the risk of the patient for developing Alzheimer's disease. Further proteomics analysis, and especially, for example, using GFAP, glial fibrillary acidic protein, can give us the microgliosis in the brain, further staging the disease. And of course, we have many metabolomic studies in AD, for example, we can say phospholipids and stuff like that, and see further these biomarkers and staging again and diagnosing correctly disease. So the topics we have put up together with my team to include in this module include biomarker requirements, classification, the criteria for evaluating potential biomarkers. We will be talking about predictive biomarkers, pharmacodynamic biomarkers, prognostic biomarkers, and diagnostic biomarkers. The practice of sample collection and qualitative control for a biomarker study in conjunction with epidemiological studies, including design, analysis, and more, mainly focusing on the pre-analytical and analytical procedures that are needed to develop biomarkers. Of course, we will discuss ethical and legal concerns, how are cohorts, for example, being chosen for developing and evaluating biomarkers. We'll go also in details of biomarker analysis tools, including flow cytometry, legal binding, DNA arrays, gene expression arrays, and proteomics. We'll discuss different biomarkers in diseases, including cancer, autoimmune disease, and neurological disorders. And finally, we will also discuss biomarker in drug discovery in animal models. And like I told you before, we're going to see how this drug involvement in biomarkers development, the use of biomarkers in drug development, and how side effects, ligand binding, and all these procedures will be affected. We hope, this is the aim of the team, that this course will enable students to develop a critical understanding of the development and validation and use of biomarkers in biomedical research. We hope that by the end of the module, like 2 years, the students will be able to critically discuss the choice of biomarkers for a defined medical research question and appraise practical issues relative to measurement in biological samples. They will be able to critically evaluate the research data in the literature that they rely on the use of biomarkers, and with emphasis on the evaluation of the translational value of these findings. Understand how new omics methodology, this is really, really important, can be applied to assist the discovery of biomarkers. Understand the steps involved in moving new biomarker discoveries to the clinical use. And of course, last but not least, appreciate the potential of biomarkers in drug development. So I hope I have described this module sufficiently enough to understand it. And thank you very much for your attention.
Hello then, welcome here. Thank you for this, for organizing this event. Dimitris Thanos, Mika Magura, and the people from MEAP. So I'll be presenting the Applied Omics module. And this is the module organized by these 3 people in academic order here at BFA, rather than alphabetical or contribution, let's say, to the preparation of the material, already prepared and submitted to the app. So applied omics and omics in general you heard about. This is one of the hottest, let's say, fields in biomedical sciences in recent years, and it is the field of biomedical science that utilizes molecular biology and biochemistry techniques. And next generation sequencing is the most prominent of that. So this facilitated these studies. We could study DNA and RNA and proteins and lipids before. However, next generation sequencing and novel technologies that drive this field in recent years have allowed us to do that in a large scale and in a scale that we couldn't do this before. And this is why it has revolutionized these studies and these fields. And you can use these approaches to study cells, to study tissues, to study organisms, to study those under homeostasis or in disease. And it is a whole family of sub-expertise, sub-fields, let's say, genomics, proteomics, transcriptomics, etc. More recently, phenomics, and there's going to be more omics, I'm sure, in the future. So, okay, so what are the trainees expected to get from this module? And after they have completed it, they will be able to describe the different categories, at least the most common ones used in basic science, but also in diagnosis and clinical research. They will be able to distinguish the different laboratory technology that is being utilized for each of these categories, and of course to kind of understand what the value of these results and the methods to analyze and how you evaluate and how they become useful, because it's always nice to get a large list of genes, but how you interpret these results is also very, very important. So, sorry, going backwards. So the individual subjects that we're going to touch upon, I already mentioned, it is genomics, epigenomics, proteomics, transcriptomics, metabolomics, and phenomics and metagenomics. That is also an emerging field with great interest, given the importance of the microbiome in various diseases, in several diseases. So the textbook that we have proposed, and I know that it's been already obtained from MEAP, is from a compatriot of ours, Dr. Stylianos Adonarakis, which is medical and health genomics, and it touches several of the fields that we're going to discuss, but also will provide literature reviews, research articles, and other textbooks. And as we go on and move forward with this graduate program, we'll enrich the literature. I'll stop here. I don't know. Slide of mine. Yep. Thank you.
So good afternoon from my side too. I'm Aristotelis Adriano, and I would like to extend my thanks for being part of this fantastic program. And the name of our module, the title of our module is Computational Biology and Applied Statistics for Precision Medicine. And as the title suggests, it aims to provide foundational knowledge on the instrumental role that computational approaches hold in the area of modern biological research, and particularly in the novel paradigm of precision medicine. Precision medicine represents an emerging interdisciplinary area that envisages diagnostic evaluation and therapeutic support by taking into account the individual genetic basis, that in the form of either vulnerabilities or immunities, but integrates to that information that has to do with lifestyle or broader environmental factors in order to optimize efficacy of the intervention. The biotech and nanomaterial revolution has provided the ground for numerous fantastic monitoring technologies, which in their own hand, they form distinct digital data modalities. So in this sense, precision medicine represents an inherently digitized area, which is characterized by a continuous explosive increase of the volume of generated data, of the heterogeneity that these data hold, and of the multiplicity that of the complexity that these data possess. So mastering all these forms of complexity requests insightful application of computational approaches and mathematical tenets from all fields of mathematics, but particularly of the area of statistics, with the scope to help the efficient design of studies and to render a sound and insightful analysis of all these big biodata. Mathematical modeling on its own promotes the in-depth mechanistic study of the diseases and pathologies that are to be investigated, and helps the refinement and the identification of the opposite strategies for drug research and development or in routine patient care. So in this module, students are expected to assimilate crucial concepts regarding the instrumental role of computational approaches in precision medicine. They will familiarize themselves with fundamental statistical concepts and methodologies in order to build robust studies, to be able to perform sophisticated integrative omic analysis with the aim to support either therapeutic stratification or even personalization, which means that we stratify our patient again and again in different patient strata as disease evolves, and to promote drug discovery. They will also learn to interpret and validate the obtained results using different tools of statistical analysis, both in a rigorous statistical fashion, but also mechanistically insightful. They will finally explore the impact of artificial intelligence, which is the hype of those days, in the form of different machine or deep learning methodologies, or even large language models in the areas of computational biology and precision medicine in particular. The course will emphasize in key computational aspects around systems biology, algorithms of molecular biology, statistical analysis and modeling, application of statistical software in precision medicine, environments like R or Galaxy, and of course, applications of artificial intelligence in the field of biomedicine. I have on purpose avoided the use of mathematical formulas because I know that mathematical formulas are notorious for intimidating students, and this is exactly what we try to avoid. We want to make students love mathematics and understand that mathematics are instrumental to simplify their lives and make them gain deeper knowledge. So with that, I would like to thank you all, and I hope that we will have success in this fantastic program. Bye-bye.
Hello, my name is Dr. Ansu Mikalopoulos, and I'll present the module named Bioinformatics in Precision Medicine, where the coordinator of that module is Professor Academician Dimitrios Thanos. Basically, what we see here is an overview of what we will discuss. It's the first thing is the omics technologies, the big data that they produce, the bioinformatics tools that analyze those big data, and how all this can be applied in disease. Finally, we will mention the fact that all this work is performed in an interdisciplinary environment, and finally, we will mention the fact that all this will also have some practicals for the students. So, the first thing we want the students is to understand the technologies, particularly omic technologies, such as genomics, epigenomics, transcriptomics, proteomics, metabolomics, etc., that produce data. And most of these data are huge in size, especially omic data. And so the second thing is they have to understand the different types of big data, such as the omic data, clinical data, environmental data, lifestyle data, etc. The next one is the fact that they have to understand that all these data are stored in databases. Those data can be stored in public databases, and then the students have to learn how to retrieve selective data from them. And they have to learn how to construct their own database to store and retrieve their own data. So the basic idea of the bioinformatics tools is to extract information from these big data that I mentioned before. I don't know if I'm an expert or how to distinguish between data and information, but I'll give you an example. Data is the fact that I drive with 160 kilometers per hour. The information is that I will get a traffic ticket. So we need to extract the biologically relevant information out of this tsunami of data that are produced by various technologies, especially omics technologies. And for that, artificial intelligence, machine learning and deep learning will assist us to perform both unsupervised learning, for instance, clustering can be used for subgrouping patients, or supervised learning to be able to classify distinct cases, disease versus health conditions, or regression, where we can predict numerical values. We will also learn graph theory-based algorithms for visual representation of protein-protein interactions and gene regulatory networks. All this will allow us to perform pattern recognition and pattern discovery, and eventually predictions. So once people have a fair, fair grasp of what those tools can do, we will apply them in disease. We will try to identify biomarkers that will allow us to predict the genetic predisposition of various people to diseases, or allow us to form an early stage diagnosis of a disease, or we can predict the outcome of this disease prognosis, or to we can use biomarkers to monitor the treatment response. Students will have to realize that precision medicine is an interdisciplinary field where biologists, bioinformaticians, and clinicians sit side by side to fight disease, to cure disease. So bioinformatics is what bridges the gap between basic research and clinical applications. Or if you want, it translates precision medicine from the laboratory to the clinic. So you can see here that what I imply is that that there are applications both in research and in routine. And finally, our students will have a hands-on experience. They will they will use those protocols and methodologies in order to search for real data, real-world data in databases, and they will process those data using bioinformatics tools. Thank you very much.
Hello, good evening. Yes. Okay. Hello, everybody. Tonight I will present you the bioethics and clinical trials module on behalf of Dr. Giulacchi. Oh, no, I don't think that this is my slides. I sent you a different slide. It's okay. Well, never mind. Well, I would like to make an introduction. You said, okay. That's funny. Well, the completion of the Human Genome Project in 2003 changed our understanding on clinical medicine and changed our understanding on clinical medicine, and that is considered as a cradle of precision medicine. And by saying precision medicine, we mean this unfolding aspect that takes into account the disease, treatment, and prevention, and integrates all the, apart from the genetic, except from the genetic, Also, the lifestyle and environmental factors. All these factors, that is what we call precision medicine, and small lines. But what we have to focus on is that I have planned something different, and I would like to make this. It's okay, never mind. So, all the data we're obtaining from the omics studies, all these big data, are integrated with the phenotypic data, with the environmental, with the demographic, and with the epidemiological data. And these can lead, by the use of a computational and sophisticated bioinformatic analysis, to the identification of novel biomarkers that can have either diagnostic or prognostic value, or biomarkers that could have a predictive value in response of novel treatments or older treatments, as well as they can reveal eligible druggable targets for not only repurposing of older treatments, but also for development of novel treatments. So this strategy can yield useful information that can shift the treatment plan from the conventional one-size-fits-all plan to a more personalized plan that takes account the individual's variability, and that ensures us that we can identify the optimal care for each patient that will ensure the minimization of adverse events. However, all novel therapies and all repurposing starts can materialize only if we evaluate them and validate them, and that can become only within clinical trials. And well-designed clinical trials. And in the era of genomics, clinical trials are very important. And within this module, we're going, the aim of this module is to explore the ethical, legal, and practical dimensions of clinical research in the era of precision medicine, to gain expertise in understanding, designing, and interpreting clinical results, to learn how to translate the molecular findings into meaningful and impactful human science. The key highlights of this module include understanding the role of human data in advancing precision medicine, analyze real-world dilemmas in clinical trials from informed consent to data privacy, and complement and interrogate the complement molecular work with critical knowledge of clinical study design and real-world data. The learning objectives of this module include bioethics. And by saying bioethics, we are going to address the foundations of ethical principles in human studies and the need of balancing innovation and patient safety. Furthermore, we are going to address the various clinical trial frameworks, the regulation and legislation governing the conduct of clinical trials, as well the ones that regulate the approval procedure, which is a field that it's being increasingly complicated these days. Furthermore, we're going to address the major innovative study designs, such as the adaptive designs, like the basket clinical trials or umbrella clinical trials, as well as the biomarker-driven studies that link molecular insights in the clinical population. Finally, we were going to see the non-interventional studies, the observational studies and real-world data collections, and leveraging real-world evidence to inform precision medicine. the completion of this module, by the completion of this module, we expect that the trainees will equip themselves with the tools needed to lead ethical, impactful clinical research that will allow them to be at the forefront of designing studies to revolutionize patient care. Thank you very much.
Hello. It's a pleasure to present to you Module 6. First of all, I'm an immunologist. I'm a researcher, professor level at the Center of Clinical Research, Experimental Surgery and Translational Research of the Bioacademy. And I will be teaching this module with my colleague Dimitra Magra and Ioanna Galani. So this module has to do with novel therapies towards clinical application. It's a very important and very interesting module, as it's going to drive the students through the various different therapeutic modalities that exist, the drugs. And that includes conventional drugs as well as emerging novel drugs, and for which we will be discussing the methods of development, the mechanism of action, the need for precision, and how many of these are increasingly positioned with precision, and what can be positioned with precision and what can't. And it will be guided through the students through small molecules, the most traditional drug entities that we know of, that includes aspirin, statins, cortisone, chemotherapeutic drugs. Also, we will be discussing peptides, like drugs like insulin, as well as the most recent GLP-1 agonists, Ozempic, or these ones that are making all the noise as anti-obesity treatments at the moment. We'll be discussing proteins. Again, these all belong to the category of the most conventional, more traditional drugs that we know of. Proteins include erythropoietin, human growth hormone, interferon, for example, and so on. And then we'll be discussing the most emerging and novel therapeutic. That includes monoclonal antibodies like the blockbuster TNF drugs, Humira, Remicade, Enbrel, which are making all the sales in big sales in the drug market, as well as others that are used, for example, anti-VEGF, like Avastin, that is used for cancer treatment. We'll be also discussing nucleic acid-based therapies, a very broad category. On the next slide, a few things more about that, as well as cell therapies. And these are therapies, as you can see in this schematic, of increasing size, complexity, but also excitement in terms of what we can achieve. Now, a very important, as I said, we will be focusing more and spending more time during the course on the most novel and most interesting and exciting developments. And that includes engineered proteins and monoclonal antibodies, which these days are going through a process of increased refinement and specificity through structural prediction methodologies, including AI approaches, and increased and continuously improved protein design for achieving better pharmacological properties, better mechanism of action, and specific mechanism of action, less immunogenicity, a broader therapeutic window, and so on. And of course, and what is particularly important for this course is that protein engineering is a key approach for achieving specificity and precision in terms of treatments. We will be also going through what we call therapeutically functional nucleic acids, a very broad range of drugs, including siRNAs, small interfering RNAs, small RNAs, by which we can target gene expression and knock down any protein of interest in the genome. Same thing for antisense oligonucleotides, longer RNAs, aptamers that can bind proteins, that are still nucleic acids, as well as
nucleic acid used for gene therapy approaches and vaccines. And when we talk about nucleic acid-based approaches, we also need to talk about delivery, because most of them, in most settings, we need delivery approaches that can, again, take these cells, therapeutic modalities with specificity to the tissue, to the cell, and to the site of interest. And methods of delivery, we will be going through methods like liposomes, cationic carriers, polymers, dendrimers. We'll be explaining this in the course, as well as attenuated viruses that can inject DNA within cells for different purposes. And we'll also be discussing vaccines, traditional vaccines and vaccine platforms, including the type of vaccine like protein unit vaccines or vaccines based on live attenuated or inactivated viruses, the most traditional approach to viruses, up to the stage of the most recent developments in terms of vaccine technology, including mRNA vaccines, the ones we heard a lot during the COVID times. And finally, we will be discussing cell therapies, a new category of therapeutic modalities, the most complex one, but as I said, the one that is most exciting. And that has different forms. I just have in the slide a couple of forms that have succeeded, because it's very important, and we'll try to pass this message to the students. This is not something in the far future that will come out. It's already in the clinic, and it's already treating people. And so we will be discussing quite a bit CAR T-cell therapy. So just to explain to you very quickly what that means is we take cells out of the individual, we engineer these cells in the lab, and then we return these cells back to the individual. And these cells are engineered to target the specific type of tumor that the individual has, and then that goes and attacks and can kill cancer cells, and in many cases, many leukemias, even induce long-term remission. And a similar approach, which is coming out, is instead of killing cancer cells, killing autoimmune cells. And great studies coming out, for example, in lupus, this devastating autoimmune disease, by which you can again induce long-term remission in this disease. We will also be discussing approaches of engineering these cells through CRISPR cell therapies. And this is again very important for disease mostly of genetic basis, by which again you can take cells out, correct the defect that exists. For example, in sickle cell anemia, there was approval of one such therapy recently, and then it's been returned back to patients, with optimal effects. And in recognition of these approaches, you can see Nobel Prize being given to these approaches, to CRISPR, to the protein engineering approach, which was the Nobel Prize in chemistry this year. So this is coming out, and this is very, very exciting. So the outcomes of the course are these that I mentioned, so the students will know and recognize and understand conventional and novel therapeutics, how RNA technologies, CRISPR-Cas systems work for genome editing and genetic and engineering cell therapies as well. Designing gene therapy approaches, barriers for all these technologies, new vaccines, cell therapies, CAR T-cell therapies, and so on. But always this will be discussed from the angle of precision medicine, how this can improve treatment to patients with increased efficacy and reduced side effects. And I would like to thank you for your attention.
Good afternoon, everybody. I also would like to welcome you to this open day, presenting the master's program of Precision Medicine and Novel Therapies, the synergistic educational activity of our research institute and the Hellenic Open University. My name is Christos Ervas, and I am principal investigator here at the Center of Basic Research. The purpose of the module entitled Biomedical, Clinical and Translational Research, which I will present to you, is to contribute to the training of our next generation of biomedical scientists, while it has been tailored for the needs of students that are receiving distance learning. The module is on the third semester and is focused on promoting an integrative lab research training. This module is also prerequisite for those students that will like to do a lab-based dissertation. First, let me briefly outline how important it is for the students to receive an integrative training in the 3 types of biomedical research. Number 1 is basic research, where scientists conduct fundamental research to understand the underlying mechanisms of biological processes and diseases, and also identify the potential molecular targets suitable for intervention, which will be used to develop new therapies. Number 2 is translational research, which is the process of turning scientific discoveries from the laboratory into treatment options for patients in the clinic. This research includes preclinical studies using animal models and laboratory testing to provide proof of concept and trigger the initiation of early-stage clinical trials. Number 3 is clinical research, which requires extensive and multiple clinical trials before the new therapy is fully incorporated in the standard medical practice, so improving human health. The trainees will learn about the fundamental aspects of basic and translational research, so they can understand how disease-oriented research is conducted in the laboratory, or how preclinical studies are performed that have an impact on human health. Because the primary focus of this module is to offer wet lab training, several experimental methods and protocols will be analyzed throughout the entire module using online distance learning platforms provided by the Hellenic Open University. During the hands-on training course, the students will produce and analyze experimental data on modern fields of biomedical science, utilizing the facilities of BRFA. The subjects of the course will cover basic research, systems biology, preclinical and translational research, and omics. The wet lab training week had an intense program of 5 consecutive days, 10 hours per day, covering the top 10 lab essential techniques. So in the first day, the students will initially go through all the necessary lab rules regarding safety and good laboratory practice, but also start to get their hands dirty by learning essential microbiological techniques used in classic molecular biology. At the last part of the first day, the students will be introduced in the DNA world and how they can handle DNA utilizing a variety of methods. So the first part will be to extract from animal tissues genomic DNA. In the second day, we will continue to cover the topic of handling DNA by plasmid DNA isolation, setting up a fundamental molecular biology technique, which is the polymerase chain reaction, or PCR, and then perform a restriction digest on pieces of DNA, cast agarose gels. And then the students will run their own prepared samples on the gel and visualize DNA, see it and isolate it. Finally, the first day will be finished by implementing specialized software programs to align sequences of DNA and familiarizes themselves with the continued updated databases in the real world. In the third day, handling proteins will be the core subject of the practical training. Initially, students will extract proteins from animal tissues and then will analyze the protein sample by electrophoresis, followed by Western blotting. With the implementation of the Western, the students will be introduced on how antibodies can be used in a variety of biological assays. In the fourth day, the students will be trained in immunofluorescence, and therefore they will be able to visualize particular proteins within the cells. The visualization will be made possible by microscopy. We are going to use a variety of microscopy techniques and also, very importantly, introduce the students to how they can not only take images, but also analyze them using specialized image analysis open software tools. In the final 5th day, the students will perform one important technique, ELISA, which is broadly used both in basic research and diagnostic labs. Then the students will visit the facility of the Greek Genome Center here at BRFA and go through the state-of-the-art equipment, which is the jewel in the crown of our institute research infrastructure. Finally, the students will assess the practical course by filling specific course evaluation forms and provide essential feedback on what did work nicely and what not for their training. There are several benefits at multiple levels of the wet lab hands-on training, and I will just highlight a few of those. The students will acquire practical skills using their own hands, and that will give them the ability to be fully operational in a wet biology lab. Important, they will know how to work safely and be exposed to the idea that lab work requires patience. But I think most importantly, for the continuously changing and evolving professional world, the students will acquire a variety of soft skills. By implementing a variety of experimental protocols, the students learn to be multitasking and manage properly their time. Working in a lab involves other people, which means that students will have to implement and develop further their communication skills. One basic idea of this module, obviously, is that we want the students to obtain familiarity with the lab equipment and a variety of techniques. While implementing several protocols, we will be constantly exposed to pay attention to detail, which is an absolutely important factor to perform successfully any type of experiment. Acquiring new knowledge is an obvious benefit for the students, which they will couple it with creative skills. Ultimately, they will improve their capacity to solve problems by asking constantly the questions underlying every scientific methodological approach: why, how, where, when, what, who. In this module, we are going to teach students 3 BRFA investigators, myself bringing my research expertise on cell and tissue mechanics in the developing animal, my colleague Manolis Mavroidis, who has a strong research expertise in molecular cardiology, both at basic and translational level, and my colleague Giannis Serafimidis, who is working on stem cell biology and tissue regeneration, implementing also basic and translational research. Thank you for your time.
Hello, everybody. I am Sarandis Giakos. I'm a clinical laboratory geneticist certified by the American Board of Medical Specialties and by the European Board of Medical Genomics and Genetics. I will talk to you today about the particular module in which I coordinate, and along with Dr. Theodoros Rabias, we will teach in this exciting master program the molecular and clinical aspects of cancers. In the forthcoming 4 slides, I will not be very tiring to you. I will outline the whole process of this particular module. So in the first part, we will discuss with the students the different types of histopathological types of tumors and cancers, the different types of neoplasias, the different distribution between the different sexes. Next, we will focus on the different causes of cancer, and especially into hereditary cancers, and to the non-hereditary genomic components of cancer, which is the most important aspect of understanding cancer biology. Our basic task would be to reveal the absolute connection of cancer and the DNA as a disease. We will discuss the effects of hereditary predisposition to cancer in the genomic integrity of the cells of the bodies and their vulnerability to different types of stressors, internal and external. We also will discuss factors environmental factors that can cause cancer, like irradiation and genotoxic exposures. In the continuation of this module, we will discuss basic principles of pathology and molecular pathology, a very important approach of cancer diagnosis and prognosis, and discuss the different relationships between the stromal cells, the infiltrating immune cells that they are coming into the tumor territory and the tumor cells, the role of the tumor microenvironment in the decision of different types of therapeutic schemes, the very important role of the continuous ongoing genomic instability of cancers, which is also now being exploited as a therapeutic means and as a mean to, for example, to see the activation of the Cas-STING pathway and the generation of immune responses into the tumor, but also the flexibility of the cancer cells and the ability for them to develop different types of resistance in any type of known therapies up to now. We will also discuss the different types of telomere maintenance mechanisms that they are very important in cancer cells in order to sustain continuous growth. And these are the most common mutations in cancer, like the telomerase promoter mutations or the p53, the guardian of the genome mutation, which is a very, very important characteristic mutation of genomic instability in cancer. What we expect from our trainees to learn is to have an idea of the very basic very basic mechanism for the generation of cancer, to understand both genetic, epigenetic, and environmental causes of cancer, to understand the different parameters that lead to malignant transformation, and to understand also the different types of approaches that we have in order to report these different pathologies between the tumors. We're talking about single cell analysis in the histopathology level or the cytology level, to the high throughput analysis of tumor specimens through genomics or other omics technologies. Also, our trainees, they must be capable to report and discuss the different biomarkers that they are now currently used in the everyday clinical practice, and to describe the different types of therapies that they might be generated through these different diagnostic approaches. For example, the genotoxic benefits of cancer cells that they lack homologous recombination repair. Tumors like the BRCA1 and BRCA2 and predisposing to breast and ovarian cancers. They are also very vulnerable to inhibitors of the PALP1 molecule. So with this slide, I will finish my presentation emphasizing to the very importance of understanding that the cancer is a DNA disease. Cancer is a disease that can be hereditary, but is also sporadic. Most of the tumors are sporadic and unrelated to aging, and this relationship to aging will be also emphasized in this module. Thank you very much for your attention.
And closing this part of this session is I'm going to talk a little bit about the dissertation, just 2 slides, to emphasize the novelty of this program, and also to make like a personal statement here. It has been a lot of work to bring this program to this stage. And I have already got several awards, but the most important one was that my son, although he's a PhD in law, he congratulated me for putting effort in giving a different dimension to distance learning. And this different dimension comes from the fact that with this program, people will have the ability to also come and do work in the labs and actually also do a lab dissertation. So we're talking about this master's program, which is a 2 years, as you have already heard, 120 European accounting ECTs. It's altogether, you need 8 modules. There are 5, which is theoretical, and they are core, they're absolutely necessary. 1 laboratory that it is also core, and it is also obligatory. And then you have to have 1 dissertation, which can be either or. So we're going to talk about the schedule a little bit more. So during the first year, the first semester, you can take maximum the 2 modules, biomarkers in precision medicine and applied omics. And then in a second semester, you will have your obligatory laboratory module of computational biology and applied statistics. And then you are going to have also the core module bioethics and clinical trials. In the second year now, things start again with a core module, which is the novel therapies towards clinical application. And then during your third semester, you can either take this laboratory module, which is biomedical, clinical, and translation research, or you can take modify your third semester and take this laboratory module, which is bioinformatics for precision medicine. So by doing that, you are choosing also a different path for your dissertation. So in your first semester, you are going to have another core module, which is molecular and clinical aspects of cancer, the last one that you heard. And then if you had taken PRF 61, you are going to do a research dissertation. Whereas if you have chosen PRF 63, you are going to have a literature-based dissertation. Okay, so this is the overall schedule if one wants to do a 2-year intense course. So with that, I would like to close the event, this part of the event. Thank everybody for your contribution and your work. And then we are ready to go on for visits in the labs for all of you interested to see our labs from close and, you know, have a good look, have a close look. So thank everybody.
Θα σε δω αύριο. Ναι, ωραία.
Αλλά